around the world, and also professor of IPB University with international reputation. We are very glad that today we will hosted two prominent scientists, one from IPB University, which is one of my mentor and also my teacher and also best friend, Professor Dodi Nandika.
Thank you.
From IPB University. And the second speaker is also my current best friend from Nottingham University. And sadly, he is younger than me. But I also learned a lot from him. Especially we spend many times in UK and also in Indonesia, in Belitung Islands. So thank you again for our speakers for joining us today. But before we start, let me introduce to you all the Director of International Program of IPB University, Professor Iskandar Siregar, who will give us an opening speech. Pak Iskandar?
Dasher, I have a short movie only for the opening. Maybe the organizer can show the movie. This is about the history of IPBTalk. Please.
We are glad to present a snapshot of 57 IPBTalk events. This 57 IPBTalk is conducted in conjunction with the 57th anniversary of IPB University this year. Thank you.
Thank you, Pak Berry.
Thank you, Prof. Iskandar. So very interactive video from Pak Iskandar. And we will move to the next opening speech from our Vice Director of Collaboration and Information System, Professor Dodi Nur Rahmat. Pak Dodi, go ahead.
Thank you, Pak Berry. Good afternoon, Indonesian time, and good morning for the time of the UK. Greetings, everyone, all the way from Bogor, Indonesia. I am Dodi Ridwan Rahmat, a professor of Rice Policy and Economics, and also Vice Rector for Collaboration and Information System at IPB University. I am warmly extending my thanks for all of you for attending this event. This is the 57th IPB Talk, tribute to the closure of the Dies Natalis or anniversary of 57 of IPB University anniversary. Thank you to our rector and also director of international program for your warm support in this program. First and foremost, I would like to express my gratitude to Professor Arif Satria, Professor Dodi Nandika, Professor Bagus Mulyadi, and Dr. Berry Yuliandi. Professor Bagus has frequently support IPB through summer courses and several activities as speakers also. IPB's talk on complexity and sustainability of science is one of the first leaders of the global debate on interdisciplinary science. Also, today's topic is very interesting to be discussed. It is a vibrant topic related to sustainable development goals. Thank you all who have always joined the IPB Talk from the beginning of the 57 of the IPB Talk. I would like to thank for always helping IPB University on its 57th anniversary. Once again, I thank you for joining with us. Last but not least, I wish you always healthy and fruitful event together. Happy IPB Talk and please enjoy the discussion. Thank you and wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Prof. Dodi. And we now welcome the Professor Arif Satria, Director of IPB University, to give welcoming remark to this event. Prof. Arif Satria, do you already join us here?
It seems not yet, Pak Berry.
Okay, so shall we move on to the speaker, or we will wait for a while?
Director has some obstacle. Pak Berry, please move on to the first speaker.
Okay, so please let me know, and we will make some space in time for Pak Rector if he already available. But now let's move to the next agenda, which is the presentation from Professor Dodi Nandika with the title Termite Plastic Moving Insects. But before that, I would like to read and introduce to you who is Professor Dodi Nandika. But I think this is a very bad question because I think almost everybody in Indonesia who have a big network knows him very well. Pak Dodi Nandika is a staff in IPB University with functional status as a professor, full professor. He was born in Banten, at Banten, on 7 December of 1951, and he was also the director of research and community service management as a director in the Directorate General of Higher Education in the Ministry of National Education, which is now called Ministry of Education and Culture. And he was also appointed as a head and leader of many prestigious and national positions, such as the head of the research and development body in the Ministry of National Education, and also the General Secretary of the Ministry of National Education. And currently, he is the chairman of the Academic Senate of IPB. And he now also the current head of the division of the, I'm sorry, so I need to read first. The enhancement of wood quality, the technology, enhancement of technology for the wood quality from the Department of Forestry Product, Faculty of Forestry, IPB. And if I have time to read all of his achievement, maybe we will not enjoy his great presentation. So I think we can let him to start his presentation. And Prof. Dodi Nandika actually already spent almost 40 years of his life to conduct research and help many company and also partners to regulate the development of research in termite exterminator. But if we hear exterminator meaning very cruel, so now in the sustainability science, Prof. Dodi tried to move on to the new era. So we will call not exterminator, but sustainable management of termite. So correct me if I'm wrong, Prof. Dodi. So Prof. Dodi, it is our pleasure to welcome you to give a presentation, maybe 20 to 25 minutes. Prof. Dodi, the time is yours.
Thank you, Pak Berry, for the introduction. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Selamat siang, good afternoon everyone, friends and colleagues. My special greeting to Pak Rektor IPB University, Professor Arif Satria, and of course my best friend, Professor Dodik Nur Rahmat, quite same name with me. And of course also Prof. Bagus from Nottingham University, who also joined us today. Good morning, Prof. Agus. Hope you doing well.
Good morning.
Good morning. Let me first congratulate Professor Arif Satria for the 57th anniversary of IPB University, which we celebrate in September 2 months ago. And also, congratulations on holding IPB Talk series, which has also taken place for the 57th time. So this event, I think, is a part of the IPB's 57th anniversary, which also happened to be IPB Talk 57 event. Therefore, I believe this event has a special meaning to IPB staff member. In this regard, let me highlight. Can we move the slide, please? In this regard, I would like to highlight one of the IPB research agenda listed in the IPB University Strategic Plan for the year 2019-2023, that is research in the field of ecology. Miftah, can you move the slide, please? For the next slide. Slide number 4, I think. Slide number 4. Okay. To my understanding, under this term of agenda, IPB heavily concerned to the development of environmental pollution mitigation strategies, including to overcome plastic pollution. This is quite urgent and of course very challenging, considering that plastic pollution is planetary threat affecting nearly every marine and freshwater as well as terrestrial ecosystem. Next slide. So, as you may be aware, countries around the world now are struggling to manage current volume of plastic pollution, including Indonesia. From the poles to the deep ocean basin, marine and freshwater ecosystem are accumulating the word plastic debris. Simultaneously, the petrochemical industry announced over 204 billion US dollar in investment driven by the shale gas boom, leading to projected acceleration in virgin plastic projection. Next slide. As the implication, about 19 to 23 million metric ton or 11% of plastic waste generated globally in 2016 enter aquatic ecosystem. It is estimated that annual plastic emission may reach up to 53 million metric ton per year by 2030. This will lead to severe negative impact on our environment. Next slide. Meanwhile, we know that research on the threat of plastic pollution have heavily focused on aquatic ecosystem. There has been limited research of plastic pollution in the terrestrial ecosystem. Therefore, scientific information regarding microplastic pollution in terrestrial ecosystem is still very limited. Next slide. In fact, terrestrial ecosystem are the pathway for plastic waste to enter into aquatic ecosystem. As you can see in the graph on the right side, even if the nation of the world succeed in achieving the ambitious target in controlling plastic emission, it is still estimated that by 2030, around 53 million metric tons of plastic emission will enter the aquatic ecosystem. Next slide. Of course, this condition require a multiple approach strategy, including plastic waste management in the terrestrial ecosystem by utilizing biological agent, or what we can call as biological control. Next slide. Unfortunately, research on biological control in handling plastic in the terrestrial ecosystem is still very limited. Really et al. 2017 have indeed examined the microplastic transport in terrestrial ecosystem by earthworm Lumbricus terrestris. Of course, likewise, Mab et al. 2017 examined the role of Collembola. Collembola is the primitive insect, as you understand, on microplastic transporting into the soil. However, once again, I would say scientific information on this subject is not widely known. Next slide. Therefore, it is time for us to recall that the Indo-Malayan region is the most important termite habitat in the world. Geographically, the center or epicenter of termite distribution is in the Indo-Malayan region, in our region. Indonesia, for example, which cover an area of only 1.3% of the planet area, has a species richness of around 300 species of termite, or more than 10% of the number of termite species in the world. Scientifically, the presence of termite in our country, in our landscape, has been reported in 45 districts or cities, or in almost 10% of the total number of districts or cities in Indonesia. It's a good, huge number. Therefore, I can say that Indonesia is a termite paradise. Next slide. In fact, the history of research on termite in Indonesia is very long history. It has been started since the early 19th century under the Dutch government administration. So we have a long journey on termite research. And now we have to turn out, we have to shift our termite research focus from termite control technology to sustainable termite management. We are no longer just a termite buster, no longer as a termite killer, but just developing, but we are determined to use termite for the benefit of society and the environment, including using termite as a source of future protein, as well as using termite to control plastic pollution. Next slide. So it is very crucial for us if we start asking about the role of subterranean termite on microplastic transportation in terrestrial ecosystem. Next, we would like to know, to assess the preference of termite species on transporting the different type of microplastic particles. Next slide. Therefore, earlier this year, together with our researcher colleague from the Center for Biomaterial, LIPI, we have started research on the role of subterranean termite on microplastic transportation in 3 types of terrestrial ecosystem, namely forest land, crop estate, and urban landscape. In this research, we examine termite preference in transferring, in transporting, or in removing 3 types of plastic, namely polyethylene, polypropylene, as well as polystyrene, both through laboratory studies and through field studies. In field studies, we install wood stack to attract termite in the several locations, then build experimental station unit at the point where termite attack the wood stakes. Our experimental station was made of PVC pipe, which was buried into the ground. Into the PVC pipe, we enter the particular bedwood and, of course, microplastic particle for a particular size. Then we monitor each experimental station for 6 weeks after installation of the experimental station. Next slide. We found that subterranean termite around the station is very active in assessing, in attacking the bedwood, as well as the stakewood. And we can collect the termite specimen to the laboratory and identify the species according to Ahmad and To. Next slide. This is the spatial distribution of termite species in our experimental site. We found that almost all of the stakes, almost of the station assessed by the termite, attacked by the termite. We found that there are 3 termite species that very active to assess and foraging to our station, namely Macrothermes gilfus, Microthermes, Odontothermes, and Capithermes. Next slide. And we also found that after 6 weeks exposure to the termite in this several locations, we found that weight loss of bedwood is quite high, reaching 50%. It means that, again, termite is very active in making foraging and assessing the termite, the bedwood inside the experimental station. Next slide. However, even we have 4 species in our collection, but there are only 3 termite species very active in assessing our station. That is Macrothermes gilfus, Odontothermes japonicus, and Macrothermes inspiratus. This is the morphology that we show to you under the digital microscope feature. Next slide. If you look the termite species distribution at each experimental station, we found that Macrothermes and Microthermes is the most active termite species in reaching, in assessing, in attacking the bedwood inside the experimental station. Next slide. After 6 weeks of exposure in the field, we found that the termites in the location is quite active to remove, to transport the microplastic particles. But we also found that the polyethylene plastic is the most type of plastic that removed by termite. Even the removing of the plastic to the soil is reached to 5 to 10 centimeter depth under the soil surface. In addition, also on the other type of plastic like polypropylene, polystyrene is also removed, transport, and maybe also move to the second layer of the soil surface until the 5 centimeter depth under the soil surface. Next slide. If we calculate the percentage of weight of the plastic transported by termite, it seemed to me that almost 40 to 50% of the weight of the plastic was removing by the termite, was transporting by the termite. Include the polyethylene, polypropylene, and also polystyrene. All of type of the plastic can transport it by the subterranean termite in several locations. Next slide. Next slide. As the concluding remark, I will say that there are 3 subterranean termite species in the experimental session. That is Macrothermes gilfus, Odontothermes japonicus, and also Macrothermes inspiratus. But the most active termite species in foraging activity that is Macrothermes gilfus and Odontothermes japonicus. The subterranean termite has potential role on vertical transportation of microplastic particle in the terrestrial ecosystem maybe until 10 centimeter below the soil surface. The type of microplastic affect the removal depth and percentage of microplastic transported by subterranean termite. In addition, the proposed mechanism of movement of the plastic particle is caused by foraging, tunneling, and feeding activities of the termite. The higher rate of food consumption, the higher percentage of transportation of microplastic and vice versa. Thank you very much.
Thank you very much, Prof. Dodi Nandika. This is a very interesting talk. And even for me as a biologist, I just now know that the termite can transport plastic vertically. And this is very promising for us in the future if we can use this kind of mechanism to make a sustainable environment. And although the time for you is still available, but maybe we can use that time to put in the question and answer session later. So if you don't mind, I would like to continue to introduce our next speaker from the University of Nottingham, Dr. Bagus Muljadi, which is actually an Indonesian diaspora working as a professor in the University of Nottingham. And Dr. Bagus Muljadi graduated from ITB in Bandung. So after that, he went to study abroad in several countries. He received Master of Science and PhD in Applied Mechanics from National Taiwan University in 2008 and 2012. And in 2006, Dr. Bagus received his bachelor degree from ITB in mechanical engineering. Since 2017, Dr. Bagus has been a professor of chemical and environmental engineering at the University of Nottingham and an adjunct assistant professor at Virginia Tech, United States. He is currently the director of Indonesian Doctoral Training Partnership and the coordinator of the UK-Indonesia Consortium for Interdisciplinary Sciences, or UKICS. Prior to joining Nottingham University, he was a research associate at Earth Science and Engineering at Imperial College, London, and at Institut de Mathématiques de Toulouse, France. Bagus' highly multidisciplinary expertise resulted in high-impact publication and grants in the field of earth science, applied mathematics, bioengineering, and computer scientists. He is co-administering over 10 million pound sterling research grant, supervising 2 postdoctoral researchers and 5 PhD students. In 2019, he was recognized as an international academic with outstanding contribution. And today, Dr. Bagus will share his knowledge and study about the architectural design of smart ventilation and drainage system in termite nests. So Dr. Bagus, please entertain us with your presentation. Thank you.
Thanks very much. I was caught off guard by saying, please entertain us. But thanks very much for a very kind introduction, Pak Berry. You are truly is my current best friend. So, and I'd like to greet the honorable colleagues from Institut Pertanian Bogor. It is truly an honor to join you again today. Pak Dodi Nandika, Pak Dase, Pak Berry, Pak Iskandar, and others. Thank you very much indeed. And as you have heard probably from the introduction from Pak Berry, I am neither a biologist nor an entomologist. So today's research talk is a little bit out of my comfort zone, to say the least, really, because I was a mechanical engineer by formation, and then I continued my PhD studies in applied mechanics or applied physics, and went on doing career in research in applied mathematics and earth science. I'm now an assistant professor at chemical and environmental engineering, really. So some of you probably think I don't know what I'm talking about, and indeed truly is the case that that oftentimes I'm really scratching the surface of a new discipline. And but today's talk is something that is very interesting. I must say this is probably one of the most interesting research areas that I have dwelt into, particularly because of two things: the uniqueness of the problem and the importance of the implications, and especially in the context of Indonesian urban area. So I got a little presentation to show you. Now, today's talk is loosely titled The Architectural Design of Termites Mounds. And so the design word there is in italics, and because it is a contentious issue, isn't it? Like, because in the UK, we always been thinking whether or not biological architectures are made by design, or is it by coincidence? And that's always been the conundrum between scientific communities and trying to, in fact, biomimicry is something that not every scientist across the spectrum would agree to utilize, because it isn't clear whether architectural design of biological creatures has some genuine utility, really. But we're trying to find out based on curiosity. So before we go further, indeed, what Pak Berry had mentioned in the beginning, we are very pleased to collaborate again with IPB. Indeed, we have hold several events at ITB, including the summer courses. And then today, because of the restriction in international travels, we limit this to online activities. But I'm so looking forward to go back to Indonesia again, and especially to hold joint events again with IPB, involving not only academics, but also students. Okay, now, excuse me. Okay, so termites isn't something that I deal with every day, and it isn't something that I deal with at all, pretty much, really. But because in Indonesia, it has a notorious reputation as this wood-eating little scary creatures that work in herds, right? And well, in colonies. So you don't typically want to deal with them, and then you tend to exterminate them, right? And then especially, it is very encouraging today to hear something along the lines of utilizing termites as means of reproducing or making better use of plastics. So the first speaker was very, very, very eye-opening. Now, termites are a bunch of interesting creatures, not least because of the variety there is in the colony, but also because of the structural design of their mounds. So what you see here are typically mounds that are found in the sub-Saharan Africa or in India. I'm not sure if this one is found often in Indonesia. These are termites mounds of meter length, but these are not the only kinds of mounds. So there are termites that eat fungi or grow fungi, really. They're called termitomyces because they cannot digest the wood. So much to the contrary of what people tend to think usually, they don't really eat wood. They take the wood back into their mounds, and then they grow fungi. And then they would let the fungus or fungi digest the woods prior to them utilizing it for whatever purpose they need. So
The key here is the fungus, because the fungus, to grow, they need to be at a forest, at a very humid temperature, something akin to a rainforest in the tropical region. So that presents its own challenges, because as you can see in the bottom left figure there, so first of all, in the bottom right, if you would make a caricature out of a termite's nest, and then you would imagine a core of a termite nest being that, sub-hemisphere area there, that's where termites usually do their activities. This is mostly their living room. It's like our living room, right? And then so the termites, they are colony creatures, and then so they have queens. We call it queens, and then the queens are responsible for the continuation of the colony, and the queen is very sensitive to temperature change and humidity and CO2 exchanges, and so these are interesting things. And what you would find in African region is that if you were to measure, and these are work by people in Harvard, I think OCO, so they would measure the temperature of the external areas of the termites mounds, and then you would see in the African region it fluctuates quite one order of magnitude difference, right? So it could be sub 15 degrees Celsius in the evenings and all the way up to 30, 35. So that's like 1 degree of magnitude difference. And then so if that's the case in the center of the termites mounds, everybody would die really, or cease to operate. No, but some people measure the temperature being fluctuating of really within 1 or 2 degrees of centigrade. So the question has always been, well, why? And does it happen by coincidence? Well, it isn't coincidence, because it happens so steadily and across the spectrum of all genus or species of termites. So they're all seem to be able to maintain their core temperature so stable. And not only that, but they seem to always have this sophisticated drainage system and CO2 exchange system that allows them to ventilate. And the question is, can we, with all our wisdom as human beings, create something akin to termite architecture like this, especially in the tropical region in Indonesia, where people are so heavily reliant on air conditioning to cool their buildings. Today's talk, or the raison d'etre of this IPB talk, is sustainability, which is very encouraging, and Indonesia has to realize its potential. I think the previous slide showed that Indonesia is one of the largest colonies of termites, right? So this type of new knowledge has always been fascinating to all of us in the UK, particularly because of our interdisciplinary culture. The question is, can you extract out whatever valuable from termites built architectures and implement that in human built systems? So because growing up, I've always been so heavily reliant on air conditioning, right? So the question is, why can't we do the same thing without, termites can do this without air conditioning. So that was always been the thing that drive our curiosity. Now, so I'm going to talk to you a little bit about the background of this research, how this come about and spiked our interest. But our colleagues in France excavated 2 termites nests, and these are not the macro termites mounds like the one that I shown you in the previous slides. These are probably sub-meter length scales of the mound. So these are non-fungus growing termites. So these are found in 2 areas in African region, Senegal and Guinea, in the city of Mouekok and Kankan. And so, as you can see, the landscape of those 2 areas are different. So the Senegal is a little less green, and then the Guinea ones are a bit more green. And then so that would reflect when we do X-ray diffraction experiments as to what constitute the termites mounds. But these are typically what it looks like without being excavated, so that was protruding out of the surface earth. And then what fascinating is that biologists aren't typically interested in the fluid dynamics of termites mounds. They're interested in probably the cognitive behavior, or the foraging behaviors. But so little studies dedicated to study how do termites actually do their CO2 exchange and so forth. So what people have done in France without having contacted us was to put this under medical X-ray. So what you saw in the bottom right figures are the excavated termites nests. These are from both Senegal and from Guinea. and then they put it under X-ray CT scanners. The one that you would encounter if you want to check whether your lungs is still operating or not. So this is what they found. The yellow areas are the chambers or the void areas, the porous areas, and then the red areas are the earth areas, the one that is impermeable by X-ray, really. Okay, so and then they want to know further, how do they actually do CO2 exchange, particularly because for this type of termites, they don't have openings to the outside world. You see, what you see, something akin to opening there is actually buried underground in figure D and E. But the one that is out of the ground, that is protruding out of the ground, has no external openings. And then what happens there, right? So what we do is that we take a subset of that external wall of the termites and then put it, we brought it to Imperial College. So this began when I was still at Imperial College. And then so this is the YouTube of Imperial College. And then we take a subset of the external wall of the termites, something like 2 or 3 millimeter across, and then put it under another CT scanners or X-ray CT scanners, but the one with sophisticated resolution. So this resolution goes down to 0.1 microns or something like that. So it's really, really high resolution CT scanners. And then I'm going to show you a video that illustrates to you what I'm talking about. So this is a very short video. So you could see the yellow one being the porous area. So these are the voids, and this is the wall, the subsection of the wall. And then so it constitutes microporous areas there, right? And then the little colorful spaghettis there are the streamlines of fluid flows. And then the red color signifies the speed of the airflows going in and out those porous sections. So those are 3 millimeters across. Okay. All right, so here's what we did. We take a subsection of the external areas and put it under X-ray CT scanners. This is something that we did often as an earth scientist in Imperial College, because we were interested in extracting oil or gases or CO2 in and out the underground or the subsurface. So this is something we do often, but it's not something people do every day in biology departments. And so we took it there and then trying to find out what the hell is going on, right? And then so, okay, and then this is what we found so far, figure A and B. And then what we do with X-ray CT scanners is that you take a stack images. So there are 1,000 or 2,000 images layered on top of each other, so as to make a 3D structures like this. And then by some watershed mechanism, you are able to extract the pore out of the solid. So this is what you typically found. So termites build their nests or mounds using pellets formed by their saliva and their excrements. So they excrete their feces, and then they cement them together with their saliva, and then they pack them, they consolidate them, and then put their consolidated pellets on top of each other. So what you see as the gray area there, those are the consolidated excrements of the termites, really. And when they put those pellets on top of each other, that constructed the large pores there. Okay, so the large pores there are something that people typically think as being non-connected between one and another. Okay, they don't typically think, well, because in order to make it connected, the termite has to actively choose as a colony to do so. Mind you, that termites are mostly blind. Well, queen has a very low visibility, but most of the termite warriors, or workers, they're blind. So it's very counterintuitive to think that they design it in such a way that the big pores are connected. But long story short, that is indeed what we found. Those long, big pores, they're connected to one another, and it has great implication in the fluid dynamics of the workings of the termites mounds. So this is what we found right off the bat. And this is something I did as an applied mathematician. So once we received 3D structures of a subsurface samples, let's say carbonate rocks or whatever, we would run Navier-Stokes simulation that simulates the way fluid moves inside the termite mound. So it's just this case. For this case, we implemented in the context of something other than rocks, but termites mound. So this is what we found. What you see in the top figure, the Senegal nests, is that the big pores, they are connected, and then they form a channel continuously from the very external exterior of the termites mound all the way to the core, to the chamber of the queen. They create this really well-connected big pores. I mean, the question to us really has been, how do they know to make such a architecture like this? Because if you could see this, without those big pores, it is impossible for flow to happen from the external to the internal. Because if you take all those big pores away, the permeability, or how easy it is for fluid to penetrate through the termites, only relying on the small pores would be something close to negligible. So without all those big pores connected to one another, there's no way termites, long story short, can breathe. And then the whole colony would die. And so that has always been the case. So we run simulations that not only simulate flow, but also CO2 diffusion using Fick's second law. And then so what we found is that the great enhancement of the diffusion processes because of this big pores being connected one and another. Another thing that we did is to find out, or trying to find out, what happens on a rainy day. So because you find high waterfall rate in several areas of the African regions, and then so what happens if it rains? Because of the capillary pressures, usually the small pores would be, we call it water clogged, right? And then so when things are water clogged, you could consider it highly impermeable, so as to very unfacilitating to exchanges of CO2 and water flow. So this is indeed what happens. So the figure A there is a dry scan of the termites mounds. And then we put water, we imbibe water into the termites nests, and then so as to make it water saturated. And then we leave it for about 2.5 hours and let it dry. And then what you will find is the figure B there at the top figure. So you would see that the big pores dry first, and that happens because of capillary pressures and trapments, and well, something that I think very common to geologists, but not very something common to entomologists and other family. And then so these are the areas that would facilitate ventilation. So we did a little bit more sophisticated measurements. Other than that, we did simulations and then quantified the permeability. Permeability is actually a variable that you could quantify, aka you put number on it. And then the permeability, albeit being 1 order of magnitude smaller than when it was dry, it was enough. It was just enough to still facilitate ventilation when rainy days happens. And then within minutes, when the water are all collected in the small pores, the big pores are already ready to facilitate ventilation and CO2 exchanges. And then, in fact, you could see in the right figure there, the amount of water are usually trapped in the pore radius that is below 25 microns there. So the big channels are free, and that's what you could find out in the simulation as well. Another thing that we did is not to look at the pore structure, but we look at the solid structure. So because we're interested in the heat transfer problem, right? Not only because the pore structure is influential in facilitating heat exchange, but the grain structure also is influential. So what we did is that we take some of the samples back into our lab in Nottingham. This one, I'm already at Nottingham. And then we conduct XRD analysis there, X-ray diffraction analysis there, so as to find out what constitutes the materials of the termites mounds. And as you could see, because termite samples from Senegal and Guinea both come from different landscapes, and one is richer in clay, whereas the other isn't. So they have different heat transfer properties there. And then so what we found is further cement our understanding of the fact that termites don't just build these structures by coincidence. They indeed have something so sophisticated in mines, whether in individual mines or in the colony mine, that make this building highly utilitarian, let's say, in facilitating the continuation of the termites colony. So that's something so fascinating. Now, another thing to talk about this research, another thing also is to encourage the framework or the mindset of people working in this so as to make such work possible. So this work or this type of work generates so much media coverage globally, and we were so flabbergasted by this, really, because this was published in Science Advances, one of the biggest journal publisher in the world, and it generates interest from the New York Times, Le Monde in France, the Volkskrant in the Netherlands, Kompas actually also covered this, the Independent in the UK, World Economic Forum, and so forth, because it was truly the first time, although people have some sort of an intuition, but they're never really looking at the details of the inner working of termites mounds prior to our work. And then so this is the cute thing about how this work originates. So I used to work in France, and therefore I met Pierre Degon there, my supervisor in mathematics department. So I wasn't in biology department. And then Pierre Degon moved to the United Kingdom, and then so did I. I moved to the United Kingdom to work under Martin Blunt, which is the chair at petroleum engineering. And Pierre befriended Guy Terola, his friend in France, who are in biology department. And then I befriended Kamal and Ali, their fellow postdocs, where I was at Imperial College. And then Pierre and I maintained a good relationship, and then we start talking about what other interesting problems that we could both pursue as the next endeavor. And then so Pierre, being talked to by Guy Terolas, talked about the importance of termites' nest. And then so I brought this research to Martin Blunt, and then everybody was interested. And people generally have no constraints as to what sort of research they should do. They're very free to pursue whatever they do there as academics without having to worry about the linearity problems. And then we thought this is interesting thing to pursue. And then we pursued this without any funding in place. So we just did it using our existing facilities. And then the event generates so much media attention that the media from the UK covered this at extensive details. And then this is us, three of us, talking about it to general Imperial College students at the time. And then so this is something that I want to encourage for you watching here. I hope not only the academics, but also students who are probably watching, that the nature of interdisciplinary science is something that truly has a chance of unlocking the problems of the future, and especially international interdisciplinary projects. Pierre Degon is French, Martin is British, Kamal is Indian, Ali is Iranian, and I myself is Indonesian. So this is the type of things that Indonesia needs to really look up to and try to spur. And this is what I commend IPBTalks about, really, because fostering this type of discussion is our best chance to lead in whatever subjects Indonesia want to lead in the future. So because of that, we established the UK-Indonesia Consortium for Interdisciplinary Sciences. And this is not least because of the contribution from IPB. Pak Arif Satria has been a great friend of ours. And then along with other universities in the UK, Warwick, Nottingham University, Coventry, and Indonesian universities such as IPB, ITB, and Gajah Mada University, we inaugurated only 3 months ago this consortium. And then we have a great hope that in the future, such work would happen cross-discipline. We want to make a free movement zone within the 6 universities so that not only I can speak at ITB, but in the future, professors at IPB could freely teach at Nottingham and Warwick and Coventry and other universities across the country. So with that, I want to end my talks. I really welcome your questions, but bear in mind again that I'm not an entomologist by formation, so I'm very limited in my understanding of termites, but there's always room to learn. So with that, I close my presentation. Thank you very much for the time. I give it back to the moderator, Pak Berry. Thank you.
Thank you very much, Pak Bagus. Very enlightening, yeah. That termite have a very high knowledge for drainage and air ventilation. And again, this is new information for me, even though I'm a biologist. So this means that still many avenue to be explored in our life and our environment that can be good knowledge to be applied in our daily life, or as you might call biomimicry. So this is very good collaboration also from many different discipline of sciences, and we can see that science knows no borders and also no countries. So means only the expertise should speak by themselves, and that can make a good science. And I know that in the audience now, already many attendees have question for both Prof. Dodi Nandika and Pak Bagus Mulyadi, and we can use two different methods to ask question here. First, you can click the reactions in the bottom right of your Zoom application, so I can see whether someone raised their hands or not. And also you can type your question in the chat box, so then we can then read your question. But before we read and allow question from the attendees, let me have the privilege to ask first to Prof. Dodi Nandika, because this is very interesting topics that termite actually can move plastics. But the question, Prof. Dodi Nandika, after they move the plastic, even until the 10-meter below the surface, what happened to the plastic? Whether they can effectively make the plastic change to other substances or other forms, or it's just a movement without any change in the structure or form of the plastic?
Yes. Well, Barry, thank you very much. Can I respond, Pak Barry?
Yes.
Yes.
Please,
Thanks you. But the simple response is we have so many unanswered questions, Pak Barry, on termite ecology, termite biology, and termite behavior. So many unanswered questions, include after the plastic particle removed and transport by termite, what happened? It's a very good question. We believe that in the near future, in our research project with our colleague from LIPI, we are eager to assess it is possible for termite to not only remove, but in the near future, we there is any insect biotechnology to, let's say, to create a particular technology that some termite species can destroy the plastic, even not for their nutrition, even not for their nutrition, but because we understand that the main food source of termite is cellulosic material, not the plastic. So again, we in the first step, we of course would like to know, is there any potential role of termite to remove and to transport the microplastic particle in the terrestrial ecosystem? But we don't know. after that, we have to explore the others called the information regarding this kind of social insect. But I believe that this is our challenging, very challenging, very interesting. But again, too many unanswered questions, Pak Beri. Thank you very much.
Thank you very much, Prof. Dodi Nandika. I will read now a question from the attendees or audience. So, in the history, since ancient until now, human never wins against termite because termites still attack millions houses and millions cubic meters worldwide, although termites are also beneficial from environmental points of view. Based on this information, questions come up. One, why studies on the termite are still few? And second, is there any study done in the developing econometric model to measure economic losses or assessments of termites attacks in Indonesia? This is a question from Ibu or Bapak Ari Nafril from the Sriwijaya University. Pak Dodi.
Yes, thank you very much, Ibu Ari Nafril from Unsri. It's very interesting question, Ibu. And I would say that, and also totally agree with you when you are saying that human never win the war against termite. In Indonesia, for instance, research on termite has been conducted since early 19th century. Miftah, can you present the slide on the termite research history of Indonesia? It's a long journey, Ibu Ari Nafril, that we have to we understand that the research on termite bioecology has been started since early 19th century, when the Dutch geologist Damerman, the director of botanical garden in Bogor, trying to explore the bioecology of Neotermas tectone, the severe termite pest on teak plantation. And after that, his student, Karlshofen, also eager to trying to explore the bioecology of Neotermas tectone on teak plantation in Java. And it's very long until now. Is more than one centuries, but we don't won the war against termite. Yes, we win some battles on termite, but no, we are not won the war. Why? Many researchers reveals that termite has extraordinary ability to adapt to any changes in its microhabitat. Termite is the supraorganism that has a very extraordinary ability to adapt to the environment, including to the various control technique made by human. So I think we understand that until now we cannot totally control the termite population, but in reverse, we understand that the economic loss caused by termite attack on our breeding is increase every year, and we understand that is our challenge to work together to shift our paradigm, maybe to use termite as our biological agent to maintain our environment, including for our nutrition. Thank you.
Thank you very much, Prof. Dodi. So I hope that the answer can help us to understand more about the termite. But there is one more question before I move to Pak Bagus for the question. Pak Dodi, this is a question from Pak Dase. Since the termite may have an impact on biodegradable plastics, should we protect the termites in our house or should we eliminate the termites?
Yeah.
This is a very, how to say, tricky question, I think. Please, Prof.
Thank
Dodi.
you. Thank you, Pak Dasir. It's a very interesting question. As you may be aware, in Indonesia, we have more than 300 termite species, but only 5% that become the insect pest to our building, to our woods. So please be aware that termite, mostly of termite species is our friend. It's a very beneficial insect for our environment, for our living. Only 5%, even less than 5%, that sometimes attack our building, our house. Yes, there are some very aggressive termite, where there are some invasive termite species like Coptotermus in Indonesia and also in Japan and in Hawaii is very aggressive in attacking our wood and our building. But again, I would say that most of the termite space is the decomposer. The decomposer is very important in the nutrient cycle in our environment. So please keep in our mind that termite is our friend. Not so many termite that we have to quote unquote to mark as our enemies. Thank you.
Okay, thank you.
Thank you, Prof. Dodi. So, if I may, I would like to point it out several key points from this question and answer season that many things still unexplored about the termites. And this is probably because first, we have so many things to explore, or second, we maybe to concentrate to several things that maybe we think more important, which is I am not agree about that. So I think we really need a new scientists, young, energetic scientists that learn from senior like Prof. Dodi that spend his life for researching termite. And maybe at the beginning, many people will ask Prof. Dodi, why you study termite? What is the benefit? What is the application? But this kind of basic research and this kind of dedication to research about something just to kill the curiosity is actually very important, and at some point in the future very important to tackle many problems in our lives, like Pak Das already mentioned, the tension between the human and environment, and also other things that maybe we will experience in the future. So I want to point it out that we need young, energetic researcher that also interested to study termite. So probably some attendees here have a junior or also student. Probably we need to spread the word about the need of this kind of research in Indonesia, especially. And Pak Dodi, I will get back to you.
Can I?
Yes, go ahead, Pak Dodi.
Yes, short comment.
Yeah.
For me, too many reasons to thank God for termite.
Yeah. That's true. Yeah. I think so.
It's depend on us how we can living together with termite. Thank you.
Yes, that's a key point. Also, to be able to live together, the tension between human and also environment. So, Pak Dodi, I will get back to you later. But let me now give several questions to Pak Bagus, because Pak Dasa actually also have a question for Pak Bagus. First is, how possible that we as human can mimic the termite architect for our building? Because as we learned from your presentation, the ventilation, the drainage, and also the heat transfer and other things very sophisticated. We cannot imagine how this creature, which is even don't have what we call brain, so actually they only have what we call ganglia, or the still a primitive network of neuron, actually can beat us in this kind of things that can make us want to reconsider the term of clever in comparison between human and animal, because we sometimes think human is the most clever creature in the world, but probably no from your presentation. And I would like to add that with the fact that Pak Dodi already presented, that the termite actually transport the microplastic. Does this kind of structure or architecture actually help them to be able to transport several things, including microplastic, to their nest? And probably you have some insight about that. And the second question from Pak Dasa is, especially for the one who still study, should we stick to our study program, or should we approach the research with other study program? What do you think about interdisciplinary approach for the research? Go ahead, Pak Bagus, if you might.
Yeah,
if you may.
thanks very much. It's the preamble of your question forces me to think philosophically, and indeed it isn't possible to do this type of work without being evoked in that regard, at least in your heart. I think we as human are given the stewardship of the earth, and then it is our responsibility to take care of it. And then the problem is that when problem arises and then we find ourselves in the mess of global warming and all this climate change shenanigans, the question should be whether we should modify the world according to our needs, or should we modify our way of living according to what's best for the earth. So there's always been the two things. And as Pak Dodi had mentioned before, we haven't won our battles for millennia against termites. So probably there are some wrong ideas in our approach. And then so the question is, how could we then, well, okay, so the question is, how do we mimic the termites architecture for buildings? Well, one thing to do that is to extract out whatever works in that microstructure of the termites, and then try to be able to create a synthetic building blocks that could have at least same characteristics of the termites. So this is not an easy problem. You're not going to make a house that looks like termites' nests, because nobody wants to live in it. Well, maybe some would like to, I don't know, but I don't want to live in a house that looks like a termite's nest, right? It would be so difficult to find my children because they'll be hiding somewhere. So the idea is that not just making chambers or windows constellated in a way that would facilitate ventilation, that's not it. But the question goes deeper than that. Could we make bricks, for example, synthetic bricks that has the pore structure, something akin to the one that you observe under micro CT scanners? And so this is the importance about interdisciplinary, just trying to segue my way into the second problem there. Because without having been in touch with the earth science discipline, you wouldn't be able to even think about trying to extract the statistical knowledge about the porosity, for example, the notion of permeability for facilitating CO2 exchanges. These are something that can be quantified and well understood among the earth scientists, but not purely understood even among mathematicians and biologists. So the thing is, without that understanding in mind, you wouldn't even begin to think that way. Okay, so what we did is that we tried to make 3D structures out of those termites' nests by printing it. with some glass materials or silicon materials, and we print it. And then we try to quantify the statistics of the pores and the connectivity of the pores. Those are very important things. So whether one pore is typically connected to 2 chambers or 3 or 4, or what would be the numbers that would maximize CO2 exchange. So these are not hand-waving arguments, okay? You really quantify those numbers and then try to make bricks that looks like that. So that's one way to do that. And then interdisciplinary research is a great way to do that. In fact, in the UK, it is encouraged for people to submit those type of research. So bodies like Leverhulme, for example, would encourage funky research like this. And then academic freedom plays a big role in promoting this type of research, because if you're free as an academic to pursue whatever research you want to do, and then you're hell-bent over commercialization, then you wouldn't even think about doing stuff like this. And also want to segue a little bit into the concept of university, right? The university is supposed to promote this way of thinking and promoting curiosity. It isn't the place to produce labor, although that's the byproduct of people who can think freely. They would be able to secure their places in the labor market, but that's not the sole goal of the university. The university is supposed to promote this way of thinking. And if the question is, should we stick in our study program or should we approach research? Well, you got to be master of one thing. That's for sure. It doesn't mean that you have to be jack of all trades and then master of none. You have to be master of one thing, but then you have to get out of your comfort zone and then trying to study other things that might not be your field of expertise. And this isn't something trivial to do, because when I ventured out of my mechanical engineering studies and go to Taiwan to study physics, I would become the most stupid person in physics. And when I went to France to study mathematics as a postdoc, I would be the most stupid guy in mathematics. And then to go to earth science, I would be then the most stupid person in earth science, and so forth. But without, I mean, so nobody wants to do that. I mean, if it's easy, everybody would do that, because but the system in Indonesia makes it difficult for people to do that. So what I would encourage is that you would lower the entry barriers for students to pursue that type of endeavors, so that they would come with a different way of thinking. So should you stick with your study? Yes, for the time being, but don't stop now. I think the question is, can you implement whatever things that you're studying right now in a very, very larger context and benefit everybody in doing so? So that would be it, Pak Beri.
Yeah, thank you very much, Pak Bagus. So we will ask you other question from the audience or attendees. So has there been any buildings existed that implemented the ventilation system of the mounds? And
Yeah.
how effective is the system to cool down the buildings?
There are many kinds of termites. So some termites have openings external to their mounds. So these are the big termites, and in Africa. So these mounds extends 2-3 meters off the ground. And then so this type of termites, because, okay, the problem is this, you get different type of porosity in the termites nest. The biggest ones are the ones where the termites actually walk through, okay? So it's so sophisticated. They make it so that the diameter, insofar that if the warrior termites were to die defending the termites chamber, the termites chamber is exactly the size of that warrior termite, so that it would block it while it's laying dead. So, okay, but anyway, so these are the biggest pores, okay? So these are the ones that you mostly see, honeycomb-like structures. Now that particular structure has been mimicked by some buildings in Zimbabwe or in communities very near to the termites. So typically they're making use of the buoyancy flow effects, because it looks like a giant chimney, right? Because it has an external opening at the very top. So hot air would usually has higher buoyancy, because higher temperature would make it lighter, right? So that's why your water boil and roil in your pan, right? Because hot liquid would tend to boil, and then so they make use of this. Now, the thing about our study isn't just to study the macropores. We're studying the micropores. That's the new thing which I probably didn't emphasize good enough. So the macropores one has been tried by humankind. So they make molds that looks like that, and then so that maximizes the heat flow. But the micropores also tells you another story. So the termites not only think about macropores, but also micropores. So the microspores termites, in terms of being implemented in human building, has never been done before. So the idea is not to make another building that looks like termites' nest, but to make building blocks that looks like the way termites make building blocks. So those are two very different things, and the latter hasn't yet been widely exploited, and that's the novelty of the work. So yeah, maybe then it is time for Nottingham and IPB then to venture onto this type of research, and we could do something together about it by involving probably architecture department, and that's actually the focus of our next grant application, which we would probably submit in 2021, among with other colleagues from across the UK, Roehampton University, Heriot-Watt, and perhaps Warwick University. So yeah.
Yeah, thank you very much, Pak Bagus. We have several key points from your answer, and probably what we have to do as a scientist, like you said, that we have at least one competencies, but we should work together with other scientists, because like you said, if you talk to different people with different discipline of sciences, actually, even though you are the stupidest person in that conversation, but actually, sometimes we can have a spark in our head that will not be thinking by the other expert in that room, because we see from others' perspective. That's, I think, what happened to that collaborative project of yours. So each person contribute to what the other person cannot see as an expert, actually. And the most interesting thing that actually the mimicry of termites structure actually already been done, but this add another level because involving the micropore of the mounds. And I will get back to Pak Dodi and you again with the similar question, because this is involving also what Pak Dodi already explained about the termitomyces. So we have Professor Lisdar Sudirman from Department of Biology, which is a well-known scientist in the field of fungi and or what he call mycology. And she said in the chat box that her student actually made the termite nest in the small bottle, and the developed comb is depend on the nodule formation of termitomyces. Pak Dodi, you mentioned about this termitomyces earlier, and probably you can explain why this happened, and maybe Pak Bagus can add when you analyze the structure of the termite nest, whether you also know that this kind of thing, or explore also the nodule formation place in the termite mounds. Pak Dodi?
Yeah, thank you very much, Pak Berry. Good afternoon, Prof. Lisdar.
Good afternoon, Professor Dodi.
Thank you very much for the very tough question.
Our student,
Our student.
Yeah.
Yes, please be informed that the student, the master student under the Professor Lisdar Sudirman supervising is also my student. So we supervise the same student named Khairul Anwar. Thank you very much. Yes, Khairul have for me. Khairul has a very great job in trying to develop the thermotemesis fungus, starting from the elites of termite, starting from the elite of termite. They trying to capturing the termite in the in special particular glass bottle. So and after almost 1 year and after evaluation, the student come to the conclusion that the termite, some of the termite species, not all the termite, is able to develop the nodule and the thermotemesis fungus and other. So I think Professor is a very great job for the student to continue for his study, because thermotemesis is the one of the edible mushroom, even the most delicious mushroom in the world. Thank you very much, Prof. Lisdar.
Okay.
Thank you very much, Pak Dodi. Bu Lisdar, do you have any following question to respond?
Oh no, I just asked Professor Bagus what maybe is that the common or what is, because it's anyway the nest is depend on the mushroom of Thermitomyces.
Yes, so Pak Bagus maybe can respond directly.
Yeah, thank you very much, Professor Lisdar. I am I'm really humbled by the questions, particularly because I'm not studying this in great depth, but and also particularly because the termites that we studied aren't the fungus-growing termites. So the Trinervitermes geminatus, they are not growing fungi, so they don't grow Thermitomyces there. But I think if there's anything I can add to that would be, it is, I think, generally conceived as a great approach to study what stimuli contribute to any structure. So, okay, so let me repeat that again. So the idea is to observe biomimicry architecture the way it is. Okay, we got a problem at hand, right? So we got a problem, we don't know what's going on. So the general approach is to simulate, either with a computer or under laboratory experiment, the growth of that architecture again. And then to observe what would be the stimuli or stimulus of that architecture. So one thing could be the fungus, right? And then you reverse engineer that, and then to try to understand the way that building emerged from the first principle. So I'm not going to, because the question is, what do I think about this? Well, what I think about this is that is generally a good approach to any new unknowns. Okay, so you would try to simplify the model, you make that in a bottle, you strip it out of our other very difficult to understand variables. So you take that one out, and then you focus on one stimulus or maybe 2 or 3 stimuli that would encourage the same building to emerge from nothing. So that's typically what a good approach should be. The difference between that work and my work is that I don't do that in a bottle, I do that in a computer. Mm-hmm. So the pore structures, they're very, very simple. So it's just made out of opaque, simple structures, impermeable to anything, and then you just flow fluid through it. So yes, so I think that's a good approach, but we don't study in depth the fungus growing termites, but very looking forward to collaborate you if there's any chance to do so, and extending our work to fungus growing termites.
Okay, thank you, Professor Bagus.
Thank you very much, Pak Bagus. So we still have times to discuss more about this, probably one or two more question. But before we get this question from the audience, actually, I'm really interested to follow up what I already asked to Pak Dodi, because when we see that the termite actually transport microplastic vertically, I'm still wonder why in the first place they interested to transport this.
Yeah.
Whether they want to eat it or they need that for any part of structure or something. Pak Dodi, I
Thank you.
really interested to know.
Thank you, Pak Berry. I have additional information that may interesting for us for further discussion. As I mentioned in my presentation, I and my colleagues in our research project, not only use the different type of plastic, Pak Berry, but also different size of the plastic. And in this regard, we provide a particular size of the plastic particle, which is quite similar with the size of the termite egg. I repeat, with almost similar with the termite egg size. So I and my colleagues quite eager to explore another information, because we have some hypothesis that the termite try or prefer to transport or to remove the plastic because they thought that the plastic are their egg. As you may understand that in termite colony, in ant colony, there's a special behavior that the workers of termite, the termite worker, usually transport their egg to the another room, to the another place that more secure for the egg. So it is very interesting. The behavior of transporting the egg by the termite worker is my reason why the termite also prefer to transport the plastic particle. Thank you.
Thank you, Pak Dodi. So I would like to touch Pak Bagus again about the termite nest, because actually we cannot imagine how actually heat transfer is important for termite, because maybe the non-biologists cannot understand how this heat actually important for insects, because several insects actually use heat for several kind of things. But the heat transfer in the nest of the termite, why is so important, and how they utilize this kind of heat transfer?
So okay. So termites, so they are very sensitive to heat. This is at least what our colleagues in France has been advising us about. So typically the resistance towards heat difference is just something smaller than 1 or 2 degrees of Celsius. So typically termites queens and the colony in the center core would cease to occupy the same location if temperature varies beyond 2 or 3 degrees centigrade. So that's really is the case, as at least for this type of termites we're studying. And then you would find by previous study, a couple of years before we did it in Harvard, that indeed the fluctuation of temperatures, regardless what happens at the exterior part of the termites, is limited to within 5 degrees centigrade. So even if it's in a very arid region in Africa or next to a forest fire, the termite's core is maintaining its stable temperature and fluctuating really be beneath 5 degrees centigrade. So we know that's important. So that's not the question anymore. The question is, well, how are they able to maintain that temperature? So because the rational thinking would be, well, can we extract out whatever wisdom there are and then implement it in our own needs? So there's no question about that. They really are sensitive to temperature changes. And then, so here's the thing. So with X-ray diffraction, not only that we could get to look at into the pore structure, but we could look at the building blocks of the termites mounds. So termites at different region, although they're the same kinds, they build their nests using different materials. Some are using mainly quartz, some are quartz and clay. Now those materials have different thermoconducting property. Okay. Now, so the structure that the termites, although they are the same termites, are different in Senegal and in Guinea. So that's what's great about it. Now the question would be, like, with a very simple neurological nervous system that they have, they were able to know what material to put at what place, at what time, and in what location within the termite's nest. So you look at the structure from Senegal and Kankan, they're very different, although they're built by the same kind of termites. And both have roughly the same thermoregulation properties. So this is something that we really continuously scratch our head about, you know. And so yeah, so this is just a preliminary work, and it remains to be seen what other wisdom we could derive out of that.
Thank you, Pak Bagus. So maybe this is the final question asked by the audience or attendees. Probably Pak Dodi should answer this question because we know that there are already several companies that eradicate insects such as termites. However, it is rarely found in Indonesia. Why did this happen? Is the termite population in Indonesia still small, or lack of knowledge and concern about termites? And is the use of pesticide to reduce termite can affect the microplastic transported? Pak Dodi.
Thank you, Pak Bagus, and also Ibu Desi Anggariani for the question. I think it's very important and interesting question. Yes, we understand that in early 1990, some multinational company developed quote-unquote new technology to control the population of the termite colony. The technology we call the colony elimination system. They use the bait of termite made of some cellulosic material mixed with the very important active ingredient that can interfere the psychological of termite. That we call that they can destroy the cuticle of the termite, so the termite can die within 4 weeks because the active ingredient can transfer to all the colony member by grooming and also topolaxis behavior. They transfer the liquid food from one worker to another worker, even to the soldier and the queen of termite. Yes, but I think this kind of method of technique is not so ecological friendly because we eliminate all of the colony member of the termite. So the question is, why did this happen? Yeah, I think in Indonesia we have a lot of company that what's called propose this kind of technology. But for me, I think we have to again to shift our research focus onto how to conduct sustainable management of termite. I think that's it for the first question. The second question, and it is the use of pesticide to reduce. As you may understand that the use of pesticide or termiticide in controlling termite colony is also what to say? It's also the old technology, because the recent technology for termite control is more focusing on provide particular what you call the bio-based material that contain the special active ingredient from the for the natural product that can interfere the psychological aspect of the termite. I think it's my response. Thank you very much.
Thank you very much, Pak Dodi. And Bu Desi actually said that she's still undergraduate, so this is very promising because she already interested to ask question about termite, even though in such a young age and young career in academic or research. And we actually still have 10 minutes if we want to move on with the question, because we already reserved time until 5:30. But if no one asks more question about this, probably we can close early this 57th IPB talk. I'm searching the attendees and audiences whether someone raised their hand or click the reaction tumbles, and apparently no one asked question. And I think this is will be a perfect time to end this event today, which we learned so much about termite and also related things about them. And for sure, okay, so I think I'm beginning to age and become old, because apparently one question is still not answered. Pak Bagus, before I close this session or this event, whether one question you can answer that, can we mimic termite nest to keep man-made building cool? Because I think you already touched this, that we cannot mimic exactly what the structure is, but we can make bricks or something that replicate this. Maybe you can give a short answer about this.
Yeah. Imitation, they say, is the greatest form of flattery. So it's if you want to think about it philosophically, we think about the designer of the world as somebody or someone or something with the greatest wisdom in mind, and then so from which we derive the solution to all of our problems. So maybe nature is filled with the signatures of those solutions. So we got problems, that's the problem at hand. The problem ranges from global warming to pollution to anything that derive from our dependency on air conditioning system, which heavily rely on great energy to do so. Now the question is, where do we seek solution? So could the solution come from a termites built architectures? The answer might be, well, we're just scratching the surface. Could we mimic it in a way that we imitate it completely from start to finish. I don't think so, because we are not termites. We are not living in colony, although some people are living in colony not by choice, but we're not. We have very different social systems, and we have very different modes of living. So you couldn't make one-to-one mapping between our buildings and theirs. So I'm not going to answer this problem with answers, but answer it with another questions, really. Now it really depends on us whether we want what sort of new kind of living that we want to live in. And the problem isn't to be solved by simply biologists or earth scientists, but also social scientists. Could we make it globally or locally, so as to make a neighborhood looking like a termites neighborhood, or locally so as to make the building blocks or the bricks looking like that? So these are big questions to think about, and I hope it could stimulate more discussions. But I'm sorry not to answer it directly, but posing more problems indeed. Thank you.
Thank you very much, Pak Bagus. So without further ado, let me close this 57th IPB talk today, and I will promise you on behalf of the International Collaboration Office that this surely will not be the last probably the last in this year, maybe, but I don't know, but probably will not be the last of IPB talk. So we will wait for the future IPB talk with prominent scientists from IPB University and also abroad. For sure, I can tell you that this talk today really increased my knowledge and also broaden my perspective in the terms of termites, behavior, and also termite structure, termite nest structure. And this probably one of the best thing happened today for me. I'm really enjoy this kind of event, because usually, like we already know that one day for lecturer in Indonesia, usually we cannot have this kind of pleasure to add our knowledge, to enjoy science. Most our duties in Indonesia now currently may be structural or administrative job, and for me this talk really recreate my joy today. One of the best thing happened today. Thank you, Pak Dodi and Pak Bagus, to make me and the audience or attendees today feel happy and also have a great joy to listen to your talk.
You're welcome.
Yeah, so I also want to thank to the staff of International Collaboration Office to make this talk happen smoothly, and also to Pak Dase, Ibu Sinto, Pak Hagi, Mbak Puput, Mas Miftah, and other staff involved in this talk. And also thanks to Pak Iskandar, who already made this event become a tradition in IPB, and Pak Dodi, Vice Rector, and also Pak Arif Satria as a Rector, to support this kind of event in IPB. Thank you very much, and see you again in the next event of IPB Talk. Please forgive me if I made a mistake during this moderation of the event. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam
Waalaikumsalam.
warahmatullahi wabarakatuh.
Sudah boleh ngomong
Bagus.
Sunda ya Pak Dodi?
Oh,
Sekarang udah
ngomong Inggris.
Thank you, Pak Bagus.
ya.
See
So
you.
after
Terima kasih,
this,
Pak Dodi, saya belajar banyak. Terima kasih.
Thank you very much, Nuhun.
Pak Bagus, matur nuwun.
Terima kasih. Pak Dasa, aduh kangen nih. Ya.
Iya, kapan ke kampus lagi Pak? Harusnya summer ini ya, summer course-nya di sini ya Pak.
Iya, diajak nggak sama Pak Beri nih, Kakak?
Nah, Pak Beri itu calon dekan kita nih.
Amin, amin.